Good morning, everyone. First of all, I would like to thank Professor Uday Khosal, my lifetime mentor and my PhD guide. I feel blessed flourishing in the field of neurogastroenterology and motility under his esteemed leadership and guidance. I feel honored to be a keynote speaker at third Research Day of SGPGIMS, and I like to thank the Research Day organizing team for providing me this opportunity to talk on advances in understanding the pathogenesis of therapeutics or Gastroperisis, a disorder of gut-brain interaction. It's honor to be here, and here is my outline I will discussing in the talks today. So starting with the brief introduction of burden and pathophysiology of DGBI, connecting the dots, microbiome and gut-brain axis, and RNA therapeutics, why it is important to fix the real pathology present with gut dysmotility and diabetes, and why the cellular remodeling of the KIT positive cells, where KIT, that is the tyrosine kinase signaling, is essential for the interstitial cells of alcohol, which is considered as a major contributor for the gut motor function for pacemaking activity. And also pancreatic progenitor cells, which is a major source for the insulin producing pancreatic beta cells, and interchromaffin cell dynamics in DGBI, and further conclusion and future perspectives. And here is my conflict of interest disclosure, working as a board member of directors at Rossview Therapeutics. And let's start with the brief introduction of the functional gastrointestinal disorders, prevalence, disease, burden, and pathophysiology. So FGIDs are currently defined as the disorders of gut-brain interaction to underscore the term functional, as it has the connotation of being primarily psychologically in origin. And 50 to 60% of the patients seen by the gastroenterologist are the patients with DGBI, particularly functional dyspepsia, irritable bowel syndrome, gastroperisis, and slow transit constipation. And in terms of pathogenesis, DGBI, it is heterogeneous in nature. And as you can see here, multiple pathophysiological mechanisms are involved that interplay with each other. And as a summary, we have learned from different studies for the major pathophysiological mechanisms which involve intestinal barrier dysfunction, gut immune dysfunction, abnormal secretion of the bile acids or chain fatty acids like serotonin or other immune and metabolic mediators, visceral hypersensitivity, and altered GI dysmotility. And the studies noted that intestinal barrier dysfunction as a core pathophysiological mechanisms in DGBI. So the gut barrier integrity is very important. And also it is important that what are the factors modulating the gut metabolic health include the food we eat, the gut microbiota we have, the food is handled by the host and gut microbes, and how the immune response of the host to the luminant contents, and overall how the GI events are sensed by the brain. So the diet is the main determinant of the gut microbiota composition and the function, and it has been linked to the human health and disease. I want to show one example. So although it is very complex picture, but what it shows that those individuals who stay at home and have a relatively diverse diet at different microbiota composition and appear healthier as compared to those patients are either in the long-term facility like in hospitals or having the institutional diet. So in these individuals, the diet and microbiota correlated with increased inflammatory markers, reduced functional independence, and reduced mini mental states. I'm showing here the result of one state-of-the-art study, how the gut bacteria interact with the brain and have the impact on the microbiome gut-brain axis. So in this placebo-controlled trial, treating patients with IBS with Bifidobacterium longum improved the symptoms of anxiety and depression. And this treatment was associated with the changes in overall IBS symptoms. So when looking at the brain structure through the functional brain imaging, Bifidobacterium longum treated patients with IBS displayed lesser engagement of the amygdala region, which is responsible for controlling the depression and anxiety symptoms. So today's talk is more on the gastroparesis, and so I will discuss that in detail. So gastroparesis is a gut motility disorder, and it is defined by the patients with delayed gastric emptying in absence of any mechanical obstruction, and in the presence of the chronic symptoms like the nausea, throwing, early satiety, bloating, lack of hunger, heartburn, reflux, spasm, and abdominal pain. And in terms of the etiology, the majority of the patients, approx like 50%, are idiopathic, and we don't know the exact mechanism. But the diabetic pathology include like the 40%, post-surgical 5%, also the post-infectious. And the gold standard for the diagnosis for gastroparesis is solid phase gastric emptying test, that is known as the gastric emptying scintigraphy, and it is considered delayed when the food bolus retention is more than 60% at 2 hours or like the more than 10% at the 4 hour. And one of the important etiology of the gastroparesis is diabetes, and which accounted for almost 1/3 of all cases of gastroparesis. So what is the burden of the diabetes? So diabetes is common worldwide, and this map is showing the prevalence of diabetes worldwide. And as you can see, like 425 people worldwide have the diabetes condition, and has an increased trend is about 48% by 2045. And as you can see, this trend in the Southeast Asia is around like 82 million people are living with diabetes, and has an increased trend is about 84% by 2045. And this is the data like in USA, like the 30 million people are living with diabetes, and 84 million are at the risk of diabetes. It means that either they are undiagnosed or in the pre-diabetic condition. And trend is like that 1 in 3 adults has the pre-diabetic condition. I love this cartoon, and showing could be you, your dog walker, or your cat jogger. One of the major manifestations of diabetes is the GI complications, and it affects the gut motility across the whole GI. It reduces the motility of the esophagus, leading to the heartburn and chest pain symptoms. And diabetes also affect the gastroduodenal motility and delays gastric emptying, and which is the hallmark of the gastroparesis. Also, diabetes impairs the small intestine and colonic motility, and the patients experience constipation in most of the cases, and about like the 20% have the symptoms of diarrhea also. And hyperglycemia leads to the reconfiguration of the mechanisms controlling the motility. So what are the pathophysiological mechanisms of gastroparesis? Practically, most of the cellular pathophysiology that we learn about gastroparesis are almost based on the deep tissue biopsy studies, and have yielded important clinical insights in the pathophysiology. And these originate from the deeper tissue of the stomach wall, muscular, raris propria, and the most of the studies have been done in the antrum, but also in the gastric corpus, and more recently from the pylorus region also. And as a summary, we have learned that the gastroparesis patients exhibit loss of c-KIT, which is the marker for the interstitial cells alcohol, and KIT signaling is required for the normal development and function. And the functional loss of the ICCs were correlated with delayed gastric emptying in diabetic gastroparesis. Also, there are some studies reported the enteric and vagal neuropathy, loss of the neuronal markers, either pan-global neuronal marker like the PGP, or the markers of the specific neuronal types like inhibitory neurons and substance P-positive neurons. Other studies have noted altered smooth muscle changes, like the functional loss of the PDGFR alpha positive cells and smooth muscle cells. And it has been recently reported changes in the gut resident macrophages population, particularly loss of the CD206 positive macrophages, which is considered to be reflective anti-inflammatory function. Also, loss of the CD206 positive macrophages are correlated with loss of ICCs and delays gastric emptying. So the ICCs are recognized as the major contributor of the GI motor activity, and I will talk about this cell dynamics in detail in the further slides. So as I mentioned, loss of the KIT is associated with the gut disorders like Gastroperisis. Animal studies reported the critical role of the c-KIT in pancreatic beta cell function and controlling the insulin secretion and protection against the diabetes development. So what we have learned from these human and animal studies, ICCs and pancreatic beta cells require KIT to develop and function properly, and loss of KIT is associated with diabetes and gut dysmotility. The mechanisms linking diabetes and gastroparesis are largely undetermined, and the regulatory factors leading to KIT loss in ICCs and beta cells were elusive, which prompts us to explore the molecular mechanisms. So how the cells communicate each other for the regulatory functions. So cell choose your words wisely. One of the important cellular regulatory mechanisms and communication between the cells is mediated by the microRNA, and these microRNA are very tiny molecules. Let's walk in through why the microRNA are important in gut metabolic health. So microRNA are the small non-coding regulatory RNAs, mediate the post-transcriptional gene repression by inhibiting translation, and are the master regulators of the cell differentiation, proliferation, and apoptosis. MicroRNA dysregulation results in functional defects in the cells, which are critical in pathophysiological mechanisms. And microRNA dysregulation might be responsible for the losses of the functional activity of ICCs, smooth muscle cells, interneurons, immune cells, which are the driving forces for the contributing to the GI dysmotility. And microRNA dysregulation in these cells regulate the key pathophysiological mechanisms of gut motility disorders, not only the GI motility, but also the gut immune function, intestinal barrier function, and visceral sensation. So other reports have noted the pancreatic islets specific microRNA regulates insulin secretion and contributes to obesity-related insulin resistance. So these studies provided us impetus exploring the microRNA dysregulation in ICCs, beta cells, which might be potential pathogenic factors leading to dysfunction of these cells through specific gene target regulation in diabetic gut dysmotility. So we recently undertook major studies in elucidating the pathophysiology of the gut motility disorders, such as idiopathic and diabetic gastroparesis. We discovered some key microRNA which play important role in gut regulatory mechanisms and are responsible for the onset and development of GI dysmotility and diabetes. So to identify abnormally expressed microRNAs in ICCs in a murine model of the diabetic gastroparesis condition, we isolated the colonic and jejunum ICCs using the fluorescence activated cell sorter from healthy and diabetic mice and performed the microRNA sequencing analysis. So what we found in microRNA transcriptome analysis is very interesting. And microRNA expression profile within the colonic and jejunum ICC from diabetic and healthy mice were quite different. As you can see here, the micro-10B was the most highly expressed in ICCs from the healthy mice and were drastically reduced in ICC from the diabetic mice. Further, we've confirmed the diabetes-dependent reduction of micro-10B expression in gastric ICCs from the healthy and from the diabetic mice. So the micro-10B is highly expressed in ICC from healthy mice, but drastically depleted in ICCs from the diabetic mice. So we speculate that the micro-10B might regulate the KIT, leading to degeneration of ICC in diabetic condition. So to confirm the functional role of microRNA 10B in ICC, we generated ICC restricted micro-10B knockout mice, and hypothesizing that micro-10B knockout mouse develop GI dysmotility and diabetes in other way around as we found in diabetic mice, which have the reduced ICC and micro-10B. So we found in mice where micro-10B are deficient, in ICCs results in ICC degeneration, leading to diabetic gastroparesis and slow toilet constipation phenotypes. As you can see here, immunohistochemistry followed by the confocal imaging showed that density of the ICCs was reduced in micro-10B knockout mice tissue in deep muscular plexus of the jejunum and cervical muscle layer in the colon. And in vivo gastric and colonic motility in these knockout mice confirmed the gastroparesis and slow transit constipation phenotypes. Further, we characterized for the metabolic dysfunctions, and we found that these mice have the impaired glucose and insulin tolerant tests. Next, we validated our findings in human samples from diabetic and idiopathic gastroparesis, and we found similar abnormal pattern of microRNA-10B, KIT, and the other transcription factors like the KLF11. And blood and gastric biopsy samples from the human patients with idiopathic gastroparesis and diabetic gastroparesis were examined and compared to the samples from the healthy control. So microRNA-10B expression was decreased in the blood samples from the patients with idiopathic and diabetic gastroparesis as compared to the healthy controls. Consistent with the data from the mice, insulin C-peptide correlated with microRNA-10B expression, suggesting that microRNA-10B may also regulate insulin productions in human. MicroRNA-10B is a better predictor for diagnosing the pre-diabetes stage as compared to the A1C. Also, we got the similar patterns of the microRNA sequencing data from the microRNA-10B read as in murine diabetic and GI dysmotility model. So, and most interesting was that micro-10B level was negatively correlated with the gastric emptying scintigraphy at like the 2 hours and also with the A1C levels. So next we asked question whether the restoration of the microRNA-10B may rescue the GI dysmotility and diabetes phenotypes. So for the gain of function, we performed microRNA-10B mimic treatments. So these are the microRNA very similar with the endogenous microRNA, and we injected high-fat, high-sucrose induced diabetic mice. Also, we compared with the top medications in diabetes and gut motility disorders like the liraglutide, sitagliptin, metformin, insulin, prochlorpride. So what we found here that, as you can see, glucose, this is the glucose tolerant test area under the curve. You can see the only the microRNA-10B group, we found like the long-term efficacy as compared to the other anti-diabetic medications like the metformin, sitagliptin, and also the prokinetic like the prochlorpride. Similar pattern we found for the insulin tolerant test also. So in addition to the metabolic improvement, functional improvement in the metabolic function, we found that gut motility was also restored in micro-10B mimic injected mice for 8 weeks, like the post-injection. And while if you see here the prochlorpride, it was effective only for the 4 weeks, but not at the 8 weeks. We found a similar pattern with the gastric emptying also. So the murine metabolic and GI dysmotility data evidence the profound and prolonged efficacy of the micro-10B treatment compared to the anti-diabetic and prokinetic medications. So these treatments, we believe that it likely restores the proper functioning of the ICCs and beta cells by increasing the KIT and insulin in micro-10B knockout and high-fat, high-sucrose fed mice, which leads to the reversal of the GI dysmotility and diabetes, as you can see evident by the confocal imaging in the micro-10B injected in high-fat, high-sucrose fed mice. There was a rescue of the KIT signaling, and also there was more number of the pancreatic beta cells and number of the islet. So the relieving symptoms is not enough. So pharmacological agents that are developed based on the cellular and molecular mechanisms underlying pathologies might provide the best avenue for the future developments. So we published this study in gastroenterology, and here is the key finding of this study. So through the loss of the function study, we demonstrated the depletion of myco-10b leads to gastroperisis, slow transit constipation, and diabetes. And remarkably, restoration of the myco-10b reverses these disease states. Myco-10b could be clinically beneficial for patients with diabetic gastroperisis. So for the role of the intestinal barrier dysfunction, we recently proposed in our review article that gut barrier dysfunction as a core pathophysiological mechanisms of gut dysmotility and metabolic disorder. As you can see here, how the gut connectome and the brain connectome are linked with each other for the gut metabolic health. And the gut barrier function is intact in the healthy condition, but there is the leaky phenotype in the disease condition. So to explore the leaky gut phenotype, we created a knockout mouse model. So that is using the Cas12 system. So we say myco-10b global knockout mice to answer the two key questions, like what are the other pathologies mediated through the myco-10b in gut dysmotility and diabetes? And number second, to illustrate which specific cell types are disrupted due to the loss of the myco-10b. As you can see here, we found the leaky gut phenotype in the knockout mice, and after the 10b treatment, the leaky gut phenotype was rescued. And myco-10b treatment remodeled the epithelial cells and rescued the leaky gut phenotype, and it is evident through the Western blot analysis also. So for the most recent discoveries on the enterochromaffin cells dynamics in gut physiology and pathophysiology, we recently discussed in our latest reviews published in JNM. So as a specialized endocrine cells, enterochromaffin cells produce up like 95% of the total body serotonin and coordinate luminal and basolateral communication in the GI tract. So EC cells affects a broad range of the gut physiological process, such as motility, absorption, secretion, chemomechanosensition, and pathologies, including the visceral hypersensitivity, immune dysfunction, and impaired gut barrier dysfunction. Further, we undertook a study characterizing the functional role of enterochromaffin cells in idiopathic gastroperisis and slow transit constipation. So in a brief summary, I want to conclude this study. So what we found that enterochromaffin cells depleted mice had the reduced mucosal restricted serotonin and displayed delayed gastric emptying and impaired colonic motility. So these motility alterations were rescued by the intragastric administration of the serotonin. So furthermore, in the patients with idiopathic gastroparesis had a significant reduction of enteral enterochromaffin cell numbers and serotonin contents, and those were negatively correlated with the gastric emptying both as the 2 hours and 4 hours. So this study uncovered a potential mechanistic link between the serotonin deficiency and idiopathic gastroparesis. So our study suggests that examining the gastric biopsy for serotonin content may be a potential complementary step for the diagnosis, prognosis, and the treatment of the idiopathic gastroparesis. So BGBI research is evolving, and understanding the heterogeneity of the pathologies at the single cell level and spatial transcriptomics of the gut immune cells, endocrine cells, ICCs, and interneurons might open window for potential therapeutic options. And exploring the gut microbiota and their metabolites, crosstalk with the host and how they remodel the gut-brain axis from motility to mood. And modern approaches integrating multi-omic data from the multicentric longitudinal study might be exploited. But it is important to keep in mind that clinical outcomes are more important compared to the mechanistic parameters alone. So in the last, I want to say that what we learn about health and disease, it comes from listening and understanding the clinical history of the patients. So please listen and listen more. Patients are telling you the diagnostic ideas and pathology present with. As Sir William Osler, father of the internal medicine, said, listen to your patients. He or she is telling you the diagnosis. I want to thank Row Lab members, particularly Professor Single Row, and funding agency, particularly NIH and Rossview Therapeutics, and the collaborators from the Stanford University, and my mentor, Professor Uday Khosal, for our time supports, for the clinical insights and leadership in the field. Thank you so much for having me. Please email or tweet me for any questions. Thanks a lot.